In the bustling city of Bioville, the Signal Transduction Express was always on the move. At the heart of this intricate railway system was a signaling molecule called Signal, a small but determined traveler. Signal's journey began when it received a crucial message from a distant station. With a sense of purpose, it boarded the signal receptor locomotive, which swiftly carried it through a network of tracks. At each station, Signal switched trains, sometimes triggering amplification engines, and at other times instructing cells to take specific actions. Finally, Signal reached its destination, where it delivered the message, sparking a coordinated response in the city. The Signal Transduction Express ensured that information was passed efficiently, allowing cells to respond to their ever-changing environment in the city of Bioville.
Signal transduction pathways and phosphorylation cascade. Our characters will be Penny the protein kinase 1, Ronald the relay molecule, Annie the ATP, Paul the protein kinase 2, Penny the protein kinase 3. And the cell will be known as a classroom. A quick note is the definition of phosphorylation cascade, which is when an enzyme introduces/phosphorylates to another, which causes a chain reaction in which there are multiple phosphorylations or introductions between proteins and enzymes. Think of it like an ongoing game of tag. This starts off with Ronald, who wants to play tag, therefore asks Penny to play and a few others, and they all agree. The game has started, and Annie got picked as it to start off the game. Since she has a lot of energy, she immediately tags Penny, and now Penny is it and she tags Paul. Paul is deciding who to tag. He notices Kenny, who isn't playing at the moment, and decides to introduce himself and invite him to play. Kenny agrees, and he decides to invite some of his friends to play as well. And now everyone in the classroom is playing. After a long time of playing, they all get tired and decide to rest and play another time. Now, the way that this game of tag represents phosphorylation is starting off with the relay molecule, which activates the protein kinase 1. The protein kinase 1 then transfers a phosphate from ATP to an inactive molecule of protein kinase 2, and therefore activates protein kinase 2. And the same thing happens and it activates protein kinase 3. And then enzymes called protein phosphates catalyze the removal of the phosphate group from the proteins and make them inactive but available to be reused another time, just like they can play tag another time.
G-protein coupled. Our characters would be Gina, the G-protein receptor, Signa, the friendly signal molecule, and Cyto, a molecule inside the cell. Gina, the G-protein receptor, lives on the cell surface on a small town called Sellington. She's known for her keen senses and her ability to connect with different signals. On a sunny day, Signa, the cheerful signal molecule, arrives in Sellington carrying a message. Signa approaches Gina with the message, but it is encrypted in a special code. Gina, the receptor, is determined to decipher it. She undergoes a change in her shape and structure to unlock the code. Allowing her to bind with Signa. Together, Gina and Signa embark on an adventure inside the cell, guided by Cyto, the curious molecule. As Gina and Signa progress on their journey, they meet various cell citizens, including Enzima, who assists in amplifying the message, and Motto, who helps transport the message to the cell's nucleus. Their adventure takes them through twists and turns, each step crucial to transmitting Signa's message. Finally, they reach their destination, and the message triggers a response within the cell, resulting in a specific action like producing a necessary protein or initiating a cellular change. With their mission accomplished, Gina, Signa, and Cyto return to the cell surface, having successfully transmitted the message. The cell's inhabitants celebrate their teamwork and the vital role played by Gina, the G-protein receptor, in ensuring the cell's proper function.